Okay, now we are going to start part 5 of this session, this marathon that we have today, discussing about electricity-based imaging methods, okay? You all have heard about what we call EEG, electroencephalography. We can have EEG with low density electrodes. So this is probably 1, 2, 3, 4, 5, 6, probably something like 8 to 16 electrodes. These are multiple array electrodes that we have, 126, 256. So there are different resolution or different numbers of the arrays that we can use in EEG. What we record from each of these electrodes are labeled with FZ, CZ. These are the electrodes that we have. For each electrode that we have, we record a wave, which is the x-axis would be time and y-axis would be voltage. So we record changes in voltage over the time for each electrode. And then, as Professor Mohannad from Iraq has mentioned, we try to, even based on the information that we are recording from the surface, we try to find the location that the electricity is coming from based on the information that we are recording from the surface. And we call that source localization. So that is what we try to do with. So it's not just, we really like to just get it's not just about the surface of the brain, but it's also about the different areas within the brain as well. One of the first important things that we do with the electricity-based methods, including the invasive methods that we discussed about, that we can measure what we call sensory evoked potentials. So what we do with sensory evoked potential, we give people a specific kind of stimuli, whether it's going to be sound, touch, some visual stimuli, some flashlights, and then we record EEG, and then we give them another stimuli, we record EEG, we give them another stimuli, record EEG, and then we basically time lock. So we bring this one under this, we cut this one and bring this under this, and then make an average from whatever we have, to have at the end we have a wave that is basically time locked based on the stimuli. So based on the stimuli that we have, we make a time lock, and then we make a sensory evoked potential. And we can have different sensory evoked potentials. This is a somatosensory evoked potential, visual evoked potential, auditory evoked potential, and this one is olfactory evoked potential. The beauty of this is when we do source localization with these evoked potentials, for somatosensory, you can see that they are located in areas that make sense, or for visual, they are in the back of the brain, or for the auditory, they are in the areas that still make sense. So you can see that how. And the other interesting thing is that we have different waves. For example, we have a wave here, which is, as you can see here, this one, or this one, or the others. And what we name here, we usually name them based on the time and whether they are going positive or negative. Here in EEG, most of the time when they go down, we call them positive, and they go up, we call them negative. So it's just kind of something that people agree they're open to use. But these numbers, so they call N7T wave. And you can see that there is something like a dynamic changes over time. So the good thing about EEG is we can see that dynamic responses, and the dynamic response within milliseconds could be detected with EEG. This is something that we cannot do with fMRI. We cannot see these dynamic changes in milliseconds with fMRI. So that is the specific quality that we have with EEG. And then there are potential, just to give you an idea, this is the kind of the EEG recording. This one is fMRI, and then sometimes we just combine EEG with fMRI to have a better idea. And this is exactly what we do in our center. So we have EEG caps, we put people inside the scanner, we do both EEG and fMRI together. It's a really complex kind of study to do because of the noise and some complexities, but this is something doable. So we can combine these different methods together and have a more sort of accurate responses. But just going back to the kind of the really basics of EEG. So as I told you, what we have in EEG, we have recordings in time and electricity, the voltage, microvolt. So we are talking about microvolt, and then as you can see here, sampling for something like 100 times per second, and this is what we can record at the end of the day from different electrodes. And for each electrode, we have an amplifier, and then we can say, okay, this one is T6, this one is T5 or T3, and then we record them over time. What we are recording from EEG are basically electrical activities that are perpendicular to the surface of the skull. So if these parts are perpendicular, so 90-degree variation, that is the way that we can record the pulses. Otherwise, the things that are happening in these other directions are really hard to be recorded by EEG. So we mainly are recording the electrical activities that are related to the neurons that are going, the signals that are going towards the skull or just getting away from the skull. So we record things in these directions most of the time in EEG. And then we have many neurons that are activating, and we record the average signal from all these neurons. And if these neurons are synchronized, so whatever you have here are neural activities. If these 6 neurons are synchronously activated or are synchronized from each other, you have different types of EEG. So what we have at the end of the day in EEG is about synchrony between neurons, between large neurons that are basically perpendicular to the surface of a skull. So that is the basic physics of EEG in a nutshell in 2 minutes. So there are definitely minor details about that we cannot cover right now, but just give a sense about what does it mean when we have. So the good thing about EEG is that we are recording the actual neural activities. That is the good thing. But we are not recording one neuron. We are recording several neurons. And we are not recording several neurons. We are recording actually the synchronization between several neurons. So at the end of the day, the signal is EEG is a synchronized, or the conclusion about the level of synchrony and the type of synchrony that different neurons in a part of the brain under the electrode are doing. And we know that these signals would be different based on the level of consciousness that we have. When we have higher level of activations, most of the time we have a less kind of level of synchrony at the EEG level and the kind of the higher level of frequency. And what we do in EEG when we have a signal, with great thanks to this brilliant guy, Jean-Baptiste Joseph Fourier, we know that whatever signal, for example, EEG signal that we are recording, we can just decompose the signal into multiple signals, simple sinusoidal signals with different frequencies. So whatever we record from EEG, we can decompose that into different frequencies and see that, okay, we have, let's say, these 3 waves that are combined to make this signal. And this is not just about frequency, but also about the amplitude. For example, this one, the amplitude of 2 Hz is higher in this case, the amplitude of 11 hertz is higher. So you have higher power here. So you can see how these are the same, with the same frequency, with different power, you can make different types of EEG waves. So what we do with EEG signal, we decompose EEG signal to frequencies and power or amplitude of those frequencies. And then we make what we call a fast Fourier transform, or what we call here power spectrum. What does it mean? This is the EEG signal. Okay, this is the EEG signal. This is what we have here is different frequencies. So we know that we have lots of activation in the 3 hertz frequency or 9 hertz frequency, and then this one shows the power or the amplitude, how much power or energy that you have here in different frequencies. So this is what we call power frequency, and we can have them in different frequencies, and then we can divide them based on different let's say beta, alpha, theta, different bands. And people try to show that different bands would have different types of meanings or different types of functions in the brain. So that is something that is going to be important as well. So that is how we have at the end of the power spectrum. So we have simple EEG recording is a time series, but the power spectrum is the process of that time series to different frequency bands. Delta, theta, alpha, we call that D tab. Delta, alpha, delta, theta, alpha, beta, and then gamma, which is over 30 hertz. And there are kind of relationships between how EEG and single neuron recordings are related to each other. I'm not going to go to the details about that. The other thing that is important to know is that there is another method to record, which we call Magnetoencephalography. What does it mean, magnetoencephalography? Instead of recording electricity, we can just record magnetic waves of these electrical activities. So if I go back to this, when you have a current which is going down or up, it is not just electricity. We also have magnetic fields around that current. So we can record magnetic field instead of recording electrical fields. Okay? And that gives us another potential to be able to measure that with a method that is called magnetoencephalography. And in magnetoencephalography, we give people lots of detectors around their brain for magnetic waves. Okay? The good thing about magnetoencephalography is that it gives us a good temporal and a spatial resolution together. So this is millisecond. If we give people an auditory stimuli, they make what we call M100. M100, this is this specific wave. This positive, on average, this is positive wave. Okay, the positive average wave that we have in 100 millisecond after the auditory stimuli. And then you can see here, that is the map of auditory cortex in both sides. And this map, if you have this map, you can have this map every milliseconds here. So the interesting part is that you can even show the brain and show that, okay, the signal is here and then goes here and goes here and goes here. So you can follow the signal in the brain across the time, because the temporal resolution is perfect. And the spatial resolution is not too bad, but so you can detect different kind of sites across the brain, and you can measure that. So there are definitely lots of really excitements about this method. It's called magnetoencephalography. There are challenges for this method as well. So first of all, the signal that we are talking about, the magnetic signal, is in the order of femtotesla. And by femto we mean 10 in the order of minus 15 Tesla. So it is really, really low. And that makes things really hard to detect, because even a refrigerator would make something like, probably something like 10 in the order of 9 times more signal than what we are recording in Meg. So it's whatever that is happening around the device is going to be recorded, and it's really hard to detect what is happening inside the brain. The only way that you can do that, you need to make a specific room and put this Meg scanner inside that room. The Meg scanner would cost you something like $1 to $3 to $5 million, and the room by itself would cost something like $1 to $3 million. So just making a room that is totally isolated from outside, from the magnetic field of outside. So it's really hard to maintain, and it's really hard to make the room. And also the challenge is that for both MRI and Meg, we need a specific system to make them cool. So we have some coolers that are making them cool, and for these coolers we use helium. So they basically consume helium, and most of the Meg devices right now consuming something like 10 times more than MRI machines in terms of the helium consumption. And helium consumption is really high, so they are really costly devices as well. So the centers that they have the Meg device, and they are using that in the proper way, there are really limited centers across the world that are doing a really good job with Meg devices. So that's another thing that I'm not very much interested to discuss about Meg in details, because there are some excitements about Meg, but even, for example, when you go to major brain imaging centers, the Martineau Center in Harvard University, they have, for example, 7 MRI scanners, and they have 1 Meg, because they need to have, just say that, okay, we have that device. But even when they use that Meg device, there are definitely lots of challenges, so I'm not going to go to the details, but there are lots of challenges to use these devices. So you need to know that how you're going to spend your money, that $10 million, in terms of, are you going to buy a Meg device or not. But the things are exciting, especially if you are interested to the audio processing, as I told you that the first author of this book is interested. So there are different figures in this book in this chapter about audio processing in the Meg device. I'm not going to go to the details about that. But as you can see here, the magnetic field is about femto Tesla. So we are talking about a couple of tenths of femto Tesla signal. And it would be interesting for you to see that what is the intensity of Earth's gravity, the G that we have, compared to Tesla. Probably do not have time to discuss about that right now, but you can check that what is the magnetic field of the planet, the Earth that we have, compared to this is way higher than what we have in this signal. So the level of signal is really, really low. And then, it could be recorded. I'm not going to discuss about that. But the beauty is, as I told you, for example, when we give people a visual stimuli, we can see, okay, there is an activation on the back of the cortex, and then the activation would go up, go to the front part, and then it goes to the other kind of more anterior parts. So you can see that over the time, you have activation that goes from the back side to the front. So you can see the temporal dynamics of these stimuli in the brain. Okay. But at the end of the day, as I told you, this is really beautiful. That this is the beauty of the field. But how you're going to use that in a real clinical practice, at the end of the day, that is going to be another important point. And as I told you, in this course, we are really interested to talk about methods that are going to be helpful in long term for clinical work in a way that would be kind of give us new information. There are definitely lots of kind of interesting things for MEC. We can do that with kids. It's easier for being implemented for kids. But we are not still sure that how that is going to be helpful for the clinical work at the end. And then there are ideas in terms of how these things are related in terms of the synchronization. I'm not going to go to the details about that. If you are interested, you can kind of I will give you some of the kind of the papers in terms of how neurons are synchronized together and how synchronized information from EEG or MEG is going to give us information about how different neurons are connected to each other. But I have one single figure that I will show you how these things are going to basically merge to each other in one single kind of area. So that is another thing I will discuss later.